Hello everyone. In DNA replication mechanism, which is divided into three phases, that is initiation, elongation and termination, the third phase which ends replication is what is called as the termination phase, and this session is going to focus on the third phase of the DNA replication mechanism, which is the termination of DNA replication. This entire mechanism is going to be taken up in the prokaryotes and especially in E. coli, as a lot has been studied using E. coli as a model system. There have been variations found in the termination process in different bacteria, but an overview of the termination process remains more or less the same. So, what are the outcomes of this session? One is going to able to comprehend that prokaryotic genomic DNA, which is a closed circular DNA, has a terminus region which is positioned diametrically opposite to the origin of replication. So that is something that is very evident in all systems. Secondly, what has been observed is that as the replication forks moves bidirectionally, they will meet at a point, they will converge, and that converging of the two replication forks helps in terminating replication. The process of termination does vary slightly in different bacterial species. More studies will reveal how much is the variation between all the different species. Yet what has been observed is there are typical mechanism. There is a typical mechanism that is seemingly able to explain how termination is happening in most bacterial species. Of course, there is an interplay of cis-acting DNA and trans-acting proteins, just as was observed in the initiation and the elongation phases of the DNA replication mechanism. In termination as well, both the DNA cis elements and the proteins as trans elements help to carry out termination. Another molecule which has played a very important role all through replication is the DNA topoisomerases. Whether it is the initiation phase or it is the elongation phase, the DNA topoisomerases have helped all the other molecules in the replisome machinery to carry replication forward successfully, and even on to end the replication, the DNA topoisomerases seem to have a very important role. What is very important to note is this, that if the termination replication does not happen correctly, then it can lead to what is called as over-replication. That is, more of the DNA is going to be replicated than is needed. And if that happens, that can lead to genomic instability. So instead of getting two daughter cells with exactly identical copies of DNA, you may not have exactly identical copies formed, and therefore that can lead to genomic instability. Therefore, it is imperative that termination replication happens correctly, and so whatever regulation and control has to happen at this region should happen. So let us look what is the structural implications at the terminus region of the E. coli DNA. So when you observe E. coli, as it is a closed circular DNA and it has an origin of replication ori C, what has already been understood is that at the ori C, the DNA is going to unwind to form a replication bubble. And with the different ancillary proteins, replication initiates in both directions. So therefore, replication is bidirectional. One replication fork will move in this direction and the other replication fork will move in the other direction. So effectively, both of them move in an opposite direction. And as they move opposite direction, and considering that their movement is, or their speed is same or very similar, they will meet exactly at a point opposite to the origin of replication. Therefore, the proclamation that the terminus region is diametrically opposite to the origin of replication. So as this moves and this moves, they will converge at this point, and hence this becomes the terminus region. But interestingly, studies have shown that the terminus region is not concentrated only in this area. In fact, it is extending on both sides. And on studying the terminus region, what was observed is that the terminus region contains several ter regions. So these ter regions, so ter standing for termination, these ter regions are 21 to 23 base pair long DNA sequences. So each one of these is actually 21 to 23 base pair long. And these 10, and there are 10 ter regions, and interestingly, these 10 ter regions are not in the same orientation. As you observe the picture over here, you will find that these 5 ter regions are oriented differently than these 5. So the polarity of these ter regions is different from the polarity of this. So if one is facing in this direction, the other 5 are facing in the other direction. So of the 10 ter regions, 5 are in one polarity and the other 5 in the other polarity. With the understanding that the 5 of the ter regions are specifically to arrest one replication fork or to take care of one replication fork, and the other 5 are to take care of the other replication fork. So effectively, what was pointed out from having studied the ter regions is that these ter regions, 5 of them are going to take care of one replication fork and 5 of them are going to take care of the other replication fork. But effectively, both the sets of the ter regions are going to initiate or enable termination to happen. So what is the understanding? That if you have one replication fork coming from one direction, say this comes from this direction, okay, then if there is a positioned ter present over here, so what one is observing is that this set of 5 is responsible for trapping this replication fork. So it gets into a trap and then it traps over here. Since it gets trapped over here, this replication fork is no longer going to move forward. Effectively, unwinding of the DNA will stop at this point. So the ter region is ensuring that the fork that is coming from this direction is trapped at this point. Now, what is being suggested is that if this ter is not able to trap the fork here, then you have 4 other ters ready to arrest it. Effectively, if one ter is not able to stop the fork, there are 4 other ters at which the fork can get trapped. So therefore, there is a provision to be able to trap the fork at more than one region in the entire terminus region. So when you look at the other ter region, okay, so this ter region is going to allow the replication fork to move only until here. So therefore this replication fork is trapped over here and it cannot move any further. So unwinding from this replication fork will stop at this region. But if you have a replication fork that is coming from this end, this replication fork is able to move through the ter. So the polarity of the ter for the two replication forks is such that it can trap one replication fork, but it is not able to trap the other replication fork. So effectively what we are looking at is that one end of the ter region is a permissive end. So since this fork is reaching the ter at the permissive end, the ter is permitting the replication fork to move through. However, the other end is the non-permissive end. So the ter has a non-permissive end. So when a fork moves towards the non-permissive end, then it is that the fork will be trapped. The fork will not be permitted to move forward. So what we are looking at therefore is very clearly is that if this fork comes, it gets trapped. But if the other fork is coming, it does not get trapped. It is allowed to come in. So a ter region basically has a permissive end and it has a non-permissive end. Therefore, the fork polarity is being decided and the arrest is also decided. So you will ultimately have one fork getting trapped, and because this gets trapped, it stays over here. In the meantime, the other fork comes and they converge at this point. So effectively, if you see, the ter region is responsible for the two forks converging. at a particular point, and this seems to be very important for termination to happen. Another important fact that comes up is that these TER sequences have been observed to have consensus between themselves, that is, they are consensus sequences. So, several studies were carried out to look at this, and it was found that when they sequenced every TER region in the terminus, so they sequenced TER B, TER A, C, D till TER J, and they compared the sequences. So on comparing the sequences, when you look at the TER region from base pair 1 to 23rd base pair, it is a very clear indication that most of these sequences do not change, or they are not different, they are similar. So there is consensus between all the TER regions, there is similarity in the sequences, but there can be changes in the base pairs at the non-permissive end and at the permissive end. So therefore, what is being suggested over here is that every TER region's non-permissive and the permissive end can vary a little. So thereby, its efficacy to actually arrest the fork may vary. Okay. Another observation that was very clearly made is that when one observes all the TER regions, it was found that in the variable portion where the consensus is less, within that less consensus region of the entire TER sequence, one finds that at the 6th position of the TER region is a GC base pairing, and this GC base pair is conserved all through all the TERs, indicating that there may be a very important role that this conserved GC base pair plays, and hence this GC base pair is not a base pair that changes easily. So one is able to understand as we move through the session that this GC base pair does has an important role to play in termination. Now, effectively, another story that is being told by the fact that these TER regions are consensus is the fact that all the TER regions can be bound to by a protein that is called as trans. So if these are the cis-acting elements, all the cis-acting elements can be recognized by a protein that is called as TUS protein, and the TUS protein, the TUS basically stands for TER utilizing segment. So therefore, the sequence that is present in the TER is utilized by the TUS protein to bind to the TER region. So you can have the TUS binding here, you can have the TUS binding here, it can bind here, 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 here. Any of the TER sequences can be bound to by the TUS protein. So this is something that is allowed because the sequences are similar. However, the affinity with which the TUS binds to this TER regions can vary depending on the variations in the sequences of the TER regions. So, a study of the Turtus interaction, a study of the Turtus complex formed has effectively shown that a TUS can bind to the TER region, and the TUS has a domain which is a motif which is an anti-parallel beta sheet region, and it is observed that this anti-parallel beta sheet region is able to bind to the TER regions, and the efficacy with which it binds to the TER region depends on the sequence of the sequence of the bases in the TER. So this is something that has been observed. So it has been proved by various studies that the TER-TUS complex is formed, and the TUS is only going to accentuate the role of TER in termination of replication. So now one needs to understand how the interaction of TER-TUS or the formation of the TER-TUS complex helps in termination. Well, there were several mechanisms that were proposed for termination. Uh, and two main proposals were taken forward for various studies. So one of the proposal is suggesting, so as you can observe over here, this is the origin of replication. You have a fork moving towards the left, you have a fork moving towards the right, so in the opposite directions, and then they would meet somewhere at the terminus region. So these are what are called as the ter regions, and you can see the polarity of these 5 are different from these 5. So for this replication fork, the arrest will be by these 5. For this replication fork, the arrest will be by these 5, because the ter region has what is called as the permissive orientation and the non-permissive orientation. So let us take the first case scenario. In the first case scenario, if, say for example, the fork 1 arrives first. Now this can happen. Why can it happen? Because one fork may move slowly if it has had to do more proofreading. So one knows that when proofreading is more, every proofreading takes halts the replication, and the correction is made, and then the replication proceeds forward. So if this fork has to do more proofreading than this fork, obviously this fork will reach the terminus region faster than this fork. So what is being suggested is that if this fork 1 is arriving its ter region first, then this helicase, which is carrying out the unwinding, will stop at this point. Why? Because the fork is going to be trapped by this C, ter C. In the meantime, the other fork is going to arrive, and because you already have this helicase over here still, you would have a point where this helicase will converge with this helicase, or this replication fork will converge with this replication fork. So as you see, as we observed in a previous slide, the convergence of the replication fork is important for termination, and this is how they are showing that the ter region can arrest the movement of one fork and wait for the other fork to arrive to converge. That is one proposal, or that is one proposed mechanism. The other proposed mechanism is suggesting that it is not necessary that the replication fork is being trapped by the ter regions. And in that scenario, what will happen is that both the replication forks will move but meet at this terminus region. And when they meet at this terminus region, because you have the convergence of the two helicases and the replication fork, you will have termination still happening at the terminal region. So eventuality is that the Turtus can help the replication fork to stop further movement, or if it is not able to stop the movement, eventually you would have the two replication forks converging to allow termination to happen. So if this portion is looked at in further detail, on convergence of the replication fork, what is noticed is that each helicase, which is still bound to the strand to which it was bound, will continue to get translocated. And as it gets translocated, what can happen is that this helicase can start unwinding the already synthesized DNA. So as you can see, it is now unwinding the newly synthesized DNA. This can happen to a certain extent. So as it translocates to a certain extent, it can unwind the newly synthesized DNA and form what is called as a 3' flap. Having formed the 3' flap, this helicase will not remain associated for a long period of time. It gets dissociated. So you have a certain length of 3' flap being formed on both sides, both the DNA. Because you have this helicase proceeding from here, creating a 3' flap in the leading strand. You have this helicase proceeding forward, creating a 3' flap on the leading strand. So this is happening only with respect to the leading strand. And then what happens is the DNA helicase dissociates. But in the process of dissociating, what has happened is that it has left what is called a 3' flap in both the DNA. So this 3' flap, the eventuality is that the 3' flap, this portion of the DNA being single-stranded, is degraded. On the 3' flap region getting degraded, on both the DNA you have a gap. Okay, this gap is present. So the DNA pol 1 comes, uses the 3' OH of the leading strand of this DNA and proceeds to fill the gap and also remove the primer. because the DNA pol 1 also has the 5 prime 3 prime exonuclease activity, so it will, based on its processivity, continue to add nucleotides, and it will remove the primer because it has 5 prime 3 prime exonuclease activity, so it is also removing the primer of the lagging strand, okay, and completes the filling up of the gap. Same in the other DNA. The DNA pol 1 uses the 3' OH that has been present on the leading strand. Using that, it is filling the gap. It is removing the primer of the lagging strand in this DNA. And the gap is filled by the DNA pol 1. However, it is not able to seal the DNA. And so to seal the DNA ligase comes up. The DNA ligase seals the DNA. So now you have complete DNA being present here and here. However, at this portion, what is observed is that the two DNA seem to be catenated. So in the interaction of the proteins with the DNA leaves the DNA in different topologies, and one of the topologies is catenation. And in termination, what has been observed is that at the terminus region, you have the two DNA in the catenated form. So replication terminates only when this catenated DNA is decatenated. So the decatenation of the DNA is needed finally to give rise to two identical DNA. So the completion of the termination of replication is with decatenation. Although all these, that is fork convergence, 3' flap generation, helicase dissociation, flap removal, gap filling, and primer removal, ligation, all of these happen at the termination region, but it does not conclude over there. Because the DNA is catenated, decatenation is the last step that happens to allow formation of the final two daughter strands. So these are the probable mechanisms that have been suggested and is also observed to happen. Now, to understand the case of the fork polarity and the arrest of the fork, or the arrest of the movement of the fork, several studies were carried out, and after all the different studies, there were two models that were proposed. One model suggested that when you have the TER binding to the TUS, because you have the Turtus complex formed, as the fork approaches the non-permissive end, okay, so the positioning of the TUS is such on the TER, depending on the permissive and the non-permissive end, the TUS also binds to accentuate the permissive and the non-permissive end, and the TUS binding domain of helicase remains open when it binds to TER. So what happens is the DNAB comes and binds to the TUS, and when the DNAB comes and binds to the TUS is able to stop the helicase activity of DNAB. So understandably, TUS is acting as a counter helicase. So what happens is that the DNAB, which has been carrying out helicase all through, on interacting with TUS, loses its helicase activity. So it becomes inactive, and on becoming inactive, eventually the DNAB is going to dissociate from the DNA. So thereby, because the DNAB is now positioned only till here, till the Turtus complex, the replication fork does not move any further because the DNAB is not unwinding any further. So the arrest of the fork is considered to be because of the interaction of the DNAB with the TUS. So this model is suggesting that the DNAB-TUS interaction is important for arresting the fork, while the model 2 suggests that the DNAB need not interact with the TUS, and even if it does not interact with the TUS, it is able to arrest the fork. So how is it being explained over here? The explanation is this, that the DNAB having its helicase activity is going to carry out unwinding of even the TER region. Okay, now as the unwinding goes on and the unwinding has left single strands of the TER region, so we know that the sixth base pair is the GC base pair, and it has been observed that it is present in all the TER regions and the GC base pair is conserved. So what this model suggested is that the moment the GC base pair is unpaired, so the hydrogen bond between the two has been broken, Who breaks that? It is the DNA-B that breaks that. So basically, it is allowing the unwinding to go on in initiated within the TER region as well. But when the sixth base pair unwinds or the hydrogen bonding between G and C happens, the C is flipped out by the TUS, okay, and the flipped C is pocketed by the TUS. So therefore, there are amino acids present in the TUS that can bind to this flipped C. So eventuality of this is that the TUS binds to the TER more strongly or with greater affinity once it binds to the flipped C. So what is suggested over here is that the flipped C happens because of melting. Who carries out the melting? It is the DNA-B that has carried out the melting. But this C flips out and the flipped base binds to TUS. So effectively, the Turtus interaction strengthens, or the association is going to be very strong. And because the association is going to be very strong and the flipped base is bound to the TUS, further unbinding is stopped. So even if the DNA-B has its helicase activity, it is not able to unwind because of the barrier that is formed due to the TUS binding to the C. So therefore, as a barrier, the TUS is not allowing the DNA-B to move forward, and therefore the fork is arrested at this point. So in both eventualities, the fork is arrested at the Turtus complex itself, but the mechanism that is suggested is different. Here the mechanism is because of melting and base flipping, and here the mechanism is because of the TUS interacting with the DNA-B and the DNA-B losing its helicase activity. So now, from this point on, what was being studied in all research was that whether it is model 1 in all bacteria or it is model 2 in all bacteria or it is a combination of model 1 and model 2 in all bacteria or it is neither model 1 nor model 2. Okay, so several studies having been done, what was observed is that the locking of the replication fork does happen. So what is observed is that studies have shown that the TUS protein definitely binds to the TER region through its antiparallel beta sheets. So the antiparallel beta sheets have been found to observe to be binding to the major groove of the TER sequence. So it binds through the major groove of the TER sequence, and the binding varies of the TUS with the TER depending on variations in the sequence in the TER region, which was predicted earlier from the earlier studies as well. But the association dissociation dynamic studies that were carried out of the Turtus complex revealed that the antiparallel beta sheet portion of the TUS binds with the TER region through the major groove. So this interaction does happen. The second thing that was observed is that there is a region, okay, in the TUS protein that forms a pocket, okay, and the amino acids around this pocket are crucial. So you can see that there are specific amino acids that are present, and these specific amino acids are able to bind to the flipped base as proposed in the previous model. So the suggestion here was is very clear, and this was what was found out by the association and dissociation study of the Turtus complex, that when you have unwinding of DNA happening and the GC base pairing is removed to release the C flips out, and on flipping out, it enters the pocket that is present in the TUS protein. And the amino acids that form the pocket are able to interact with the C, holding the C. So this interaction is making the TER interaction with the TUS stronger, and also what happens is no further unwinding of the DNA happens beyond this point of the TER. So in this study or this fact was also substantiated by using a defective helicase, that is a helicase which could not unwind, and also by a study that did not use the helicase at all, but what they did is they carried out unwinding using different concentrations of KCl. So we all know that salt is also responsible for unwinding of DNA, or what is called as denaturation of DNA. So a study was carried out just using KCl, and because of the KCl concentration, the unwinding of the DNA does happen. On an unwinding of the DNA, it was observed that the Tertus complex association was observed without the helicase being present. Okay, so therefore this study is definitely suggesting that model 2 happens. Okay, but whether this is the only thing that is responsible for termination or not was not really very clear. But what is definitely observed is that the log domain selectively interacts with the flipped C upon strand separation. This was established by the study carried out through this research. Now, another study that was carried out using a looking at the polarity. So what was observed is that when helicase is moving towards the permissive end, okay, so when it moves towards the permissive end of the Tertus complex, then the helicase is able to dissociate the TUS. So you can see that it dissociates the TUS, and because it dissociates the TUS, it continues to move through the TUR region and carry out unwinding. So therefore, that is the explanation, and this has been carried out by using mutated TUS regions, mutated TUS proteins, and also mutated helicase. And in each case, what is observed is that if it goes to the permissive end, the helicase is able to dissociate the TUS and move forward. But when the helicase comes through from the non-permissive end, the helicase has not been able to dissociate the TUS region. And that is because the TUS interacts with the GC, the flipped C, and strengthens its association with the DNA. And therefore, the helicase is not able to dissociate the TUS. Okay, what has also been significantly observed through this study is that when you have a defective TUS, okay, then it is observed that although the TUS can bind to the TER region, it is not able to stop the helicase. But when you use the TUS which is not defective and the TUS binds to helicase, the helicase does not move forward. So what has been suggested by the studies done is that both the models are happening at the same time. That is, the TUS is interacting with the DNA helicase and it is stopping the helicase activity of DNA B. On the other end, what is also happening is that the TUS is able to lock the replication fork by interacting with the flipped C. So both the models are working together hand in hand to enable the fork arrest and to enable the further and to stop the further unwinding of the DNA by making the helicase inactive. So both the things are happening at the TER region thanks to the Tertus complex. So this is something that has been observed. Now therefore, the further thing after the replication fork has stopped and the DNA B dissociates, okay, you would definitely have the DNA pol 1 coming and completing the replication because you have gaps present at the terminus region. So all that happens. And what is left eventually is with the catenated DNA after termination. Now, understandably, during replication, all through, the DNA topology keeps changing depending on what are the molecules that are associated with the DNA. So one already observes that because the helicase keeps unwinding, you have positive supercoils formed ahead of the unwound region. So you have more overwound region over here, which is taken care of by DNA gyrase. So that begins with initiation and all through the replication along in alignment with the elongation phase, the DNA gyrase keeps paving the way for helicase to unwind further. So that is something that is already observed. What is also observed is that the newly synthesized DNA can also tend to form catenins. So these pre-catenins are being removed by another polymerase which is called as topoisomerase 3. Not polymerase, topoisomerase which is called as topoisomerase 3. However, at the termination region, when you have the two DNA catenated, what is observed to be acting is the topoisomerase, DNA topoisomerase 4. So these DNA topoisomerase 4 are able to cleave two strands. So it is class 2 type of DNA topoisomerase, and therefore it is able to cleave the DNA at these sites, allows one DNA to swivel through, and therefore eventually you will get two separate DNA molecules. So this decatenation process actually is the end of the termination process, and therefore this is a very significant thing that happens towards the to enable termination of replication. So topoisomerase 4 in most of the bacteria are responsible for decatenation step. So let us conclude. The termination of replication takes place at a region diametrically opposite to the origin of replication in bacterial DNA that has been substantiated with proof. The terminus region contains cis-acting 20 to 23 base pair long TER sequences. We know there are 10 TER sequences in E. coli, which in turn are polarized. So 5 are in one orientation and 5 TER regions are in another orientation, with the TER region itself having a permissive end and a non-permissive end. The permissive end allows the replication fork to move through, whereas the non-permissive end does not allow a replication fork to move through. So it is at the non-permissive end that the fork is trapped. The TER regions have consensus sequences, and because they have consensus sequences, all the TER regions can be bound to by TUS. The TER-TUS complex together is accentuating the fork polarity to arrest the fork and to stop the helicase. So after helicase dissociation, the filling up of the gaps is followed by decatenating the replicated DNA to give rise to two separate DNA. Thus, termination of replication is due to the dual effect of trapping the fork by the Turtus complex and binding to the helicase to stop its helicase activity. The forks meet to converge, catenate, and decatenate, thereby leading to termination. Thank you.